Good morning, everyone, and welcome to day 3 of our special AIRR Community Event, leveraging AIRR sequencing data to inform the biology of COVID-19. Today is going to be a pretty different format from the first 2 days, in that we'll have basically 3 different panel discussions. The topic that we're covering here is how COVID-19 can serve as a catalyst for the implementation of the FAIR guiding principles for AIRR-seq data. And we'll get to interpreting what that means in a little bit. But right now, I just want to remind you that we're going to be broken up into 3 panel discussions with 10-minute breaks in between. And then finally, we'll end the conference with some closing remarks from our chair, Nina Prack. So I'm going to give you a bit of background on this this 3rd day, and for those of you who might have not been around for the beginning of the conference, I'll do a very quick review of the what the AIRR community is. So that's the Adaptive Immune Receptor Repertoire Community, which began in 2015. And overall, what the community does is to develop standards and recommendations for obtaining, analyzing, curating, and comparing and sharing AIRR sequencing data sets. And AIRR sequencing data, again, has got to do with the sequences of B cell and T cell receptors and as repertoires. So that means thousands to millions of sequences. Now, the AIRR community also promotes its standards by working to become interoperative, having our standards become interoperative with other group standards for similar data, making software open source and open license and freely available, creating repositories containing published AIRR-seq data. So we have several repositories like that where they've gone into the literature, taken data, and put them into their repositories so that they are AIRR compliant. They're complying with our standards, and as a result can be easily queryable and searchable. And finally, we also, as we're doing today, hold meetings, workshops, and so forth on the progress made in basic and biomedical sciences of the AIRR-seq data formats. So today's panel discussions, why are we even having them in the first place? And so I've got
Excuse me, Jamie, I'm sorry to interrupt, but we don't see your slides.
Oh my gosh, you're kidding me. Okay.
Yeah, thank you.
Oh, yeah, so I guess I have to get out of this and go into sharing. Sorry, people. Yeah, I just had a set of slides. Share screen. I forgot to do that. Okay. So, can you see my slides now? Can you all see my slides now?
Yes.
Okay, great.
Yes.
I'm so sorry. That's really awful of me. Okay, so so here's today's you've got this in your agendas, and I just introduced the AIRR community to you. And so the story behind today's panel discussions, have to do with the AIRR community wanting to have solved problems that they saw as being important. I'm sorry, but I can't see my own slide. There we go. Okay. But they saw that they needed to solve some problems that were impeding the seamless use of these AIRR-seq data. So as a result, we actually got together, formed a community, and developed working groups, of which there are now 8, which are loose networks then of volunteers who are working on specific problems. And the thing to know about these working groups is that once their topics once their problem is solved, they can go away essentially, and they may evolve into something else, such as a long-term review subcommittee, which I'll mention in a minute. So right now, this is a list of our working groups, and I want to just give you an example of one of them, the minimal standards working group, where you can go on the website and you can check out what the group does and what their goals are for the year. But the important thing is that you can also, for instance, find in these places how to, for instance, upload your AIRR-seq data to NCBI following the AIR compliant format. So all of that is, you know, up on our website and is described. There's also publications. There's publication on these standards as well. Another one that I want to just mention briefly, another one of our working groups, is the germline database. This is germline genes that are inferred out of AIR-seq data. And this is a group that has spun off a resource, the OGRDB, which is basically a set of guidelines for getting inferred alleles to be accepted by the IMGT. And so that became actually its own subcommittee. And here's their purpose and achievements and so forth, where the OGRDB database, you can get into it and see, well, what does it take? I've got some inferred genes here. What does it take for them to become accepted by the community? Okay, so so that's pretty much, there we go. So so those, our working groups then have been, you know, functioning for 5 years now, but we've started to see that there's a larger set of groups out there working with different kinds of data. And of course, systems immunology is using kind of multidimensional large data to do analyses to describe and characterize the networks and model the networks within the immune system. So many of us began to realize that in order to do multidimensional analyses, that other groups are going to have to be doing the same things that we are. In other words, trying to standardize, and then we also need to not only standardizing their data types, but also we're going to have to create shared formats and so forth so that these data can become more, quote, interactive. So we also came to understand that for big data like AIRR-seq data to have maximal impact and therefore to have maximal value to the scientific community, the structures should be reflecting the values that we've built into the AIR community, those of collaboration, inclusivity, transparency, and data and materials sharing. So it turns out that our values are a lot like those of many other scientists. And I think that's what makes our working group so attractive. So today, our 3 panel discussions are meant to take us further into the meat of some of these issues. We built them around the FAIR guiding principles for scientific data management, that is making data findable, accessible, interoperable, and reusable. So we're going to be kind of following through with these overarching themes. And so today, again, we're going to have 3 panels that will be talking about what fairness is and what the achievements have been made by following these guidelines and principles and what their pros and cons are. Well, that'll be run by Christian Busa. And then how are database providers making COVID-19 data fair? So Bjorn Peters is going to be moderating that, and we've got several people, panelists, who have run databases themselves. And one question I've always had is, how do you even attract people to use your database and put data into it in the first place? So they'll be accomplishing that. And then finally, I'll be running this last piece on implementing policies to support data, fair data. In other words, implementation through societies, through journals and industry and so forth. Okay, well, with that, I want to move on to introduce Christian Busse, who will be running this panel on the state of FAIR 2020, the achievements, catalysts, obstacles, and risks, along with his panel members.